We're going to start our journey through the eye here. In the fall, I taught you about the connections of the extrinsic eye muscles and their innervations, etc. This is not intended to be anatomy, but of course, if you're going to talk about how the eye is moving, you have to learn again. Once again, we're going to bring up the innervations. LR6SO43, yeah, they're done with the extrinsic eye muscles and their movement of the eye. You have to move the eye about. Now, we didn't talk a lot about this in the fall because I just really wanted to emphasize the muscles and stuff, and we sort of gave very short treatment even to the eye anatomy. And we're not going to do much better here, except that I want to mention some things that a good physiologist, a good eye physiologist is aware of, things that go wrong with the eye, and if you're going to explain those things, you really need to know something about the structures that are there. For example, I put a couple pictures in this one to illustrate this for you guys today. Most people have seen someone that has some sort of problem with their eyes, and I want to try today to explain some of these in terms of what are the things there that are causing this stuff. This is not really physiology, but it's interesting, I think. I hope it's interesting and useful information about what's going on in the person's eye. Like if a person's eye is bloodshot, what is that? Where are those blood vessels? I mean, what is causing that? Is that related to, whenever you put a contact lens in your eye, where is it? Whenever you see somebody with a swollen eyelid, what's causing that? Those kinds of questions I'd like to address when you say somebody has pink eye or conjunctivitis. What does that mean? So these are the questions that I want to try to address as we work our way in and try to get you interested in eye physiology. So the first thing is, you guys already know, if you're going to close your eyes, that this is done with the contraction of a muscle called the orbicularis oculi, circular muscle that goes around your eye. It's a constrictor, and it constricts the orbit, the orbital area, and closes the eye. And you also know if you're going to open your eye, the primary mover of eye opening is this muscle in the top eyelid. The fancy word for eyelid is palpebrae, called the levator palpebrae superioris, which is connected to the upper eyelid and draws it open. Now, I didn't mention this to you guys in the fall, but there are a number of ways that you can create problems with keeping the upper eyelid open. So we know that this is a skeletal muscle, keeping the eye open, but there is also a band of smooth muscle. Here, this points to this pink area here, and this is actually slightly mislabeled. That is where the tarsal glands are, but there's also a piece of smooth muscle here in the eyelid that actually contracts reflexively when the eyelid is open, and it helps to keep your eyelid open. It's actually innervated by the superior cervical ganglia of the sympathetic nervous system. We'll come back to that in just a moment. This guy, the smooth muscle that's here in the eyelid, helps to do that. And so this is one way. Of course, you can do this with somatic problems in the oculomotor. You got a problem in cranial nerve 3's innervation here of the levator, you're going to have a droopy eye. This is where cranial nerve 3 problems can result in that. But you can also get problems that show up in the sympathetic nervous system with the eyes that are caused droopy eyes. And that's because there's not just skeletal but also smooth muscle in the eyelid. So you see somebody with the droop eye, what's causing that? Well, it could be that it's somatic stuff coming from cranial nerve 3. There's a problem with cranial nerve 3's innervation of the levator. But it could also be that there's a problem in the sympathetic nervous system's innervation of the smooth muscle, the tarsal muscles in the upper eyelid. Now, I'm going to say this, I won't ask you to remember this, but it should ring a bell for you. Those sympathetic fibers that go to that tarsal muscle in the upper eyelid pass through the cavernous sinus. Now, that is a true dural sinus on the lateral aspects of the sella turcica. You know where the dura is split? It's a pocket of blood in there. And I showed you guys where the ophthalmic and facial veins pass blood into that dural sinus. And there are lots of important things in there, like cranial nerve 3 and 4 and V1 and V2 and the internal carotid artery. There are very important pieces and parts in there, and if you get something draining from the danger triangle through that area, you can create some problems. This is one of them. This droopy eye phenomenon, not being able to control the upper eyelid. All right, well, you just started thinking about the eyelid. Now, it's true also that the eyelids, both of them actually, are responsible for producing material that sits on the surface of your eye. This material is mostly oily material. The most watery, the highest water content material on the surface of the eye actually comes from the lacrimal gland, which you've heard about before. I'm going to show you a picture of that. But I did not mention all these other glands. There are lots of glands associated with this eyelid. For example, there's a group of glands called the meibomian or tarsal glands that sit just inside the eyelids. These guys are actually sebaceous glands. Now, the reason I bring them up for a point of interest here is sebaceous glands produce an oily secretion. And sometimes those oily secretions clog the ducts that they come out of. And this is what that looks like. So, oftentimes when you see a massively swollen eye, it looks like it's swollen from the inside. This is, I always think about this when I'm having Italian food. There's a famous Seinfeld episode where George gets a calzone for George Steinbrenner. And every time I watch it, I can't help but think about this problem. I tell my wife, hey, that sounds like a problem with the tarsal glands. But it has nothing to do with it. It's spelled differently. It just sounds, you say the word a similar way. It's the pronunciation on the second syllable. It's calazion, is the way you pronounce it. You may have referred to this as a sty, which is technically not the right term for the tarsal gland infection or the tarsal gland inflammation. But anyway, I want you to see it. This is a problem with the eyelid here with the tarsal glands. Now, that's not the only kind of glands. In addition to there being true sebaceous glands, there are also sweat glands associated with the eyelids. Now, these guys tend to be very close to the exit points of the hairs, the eyelashes that come out of the eyelids. And these guys are actually apocrine sweat glands. Now, remember, there are several varieties of sweat glands. Eccrine is more watery, but these are more oily here. They're more cellular in their content. And really, the viscosity of the secretions of both these guys and these guys actually help to create a film, an oily film on your eye that helps keep it lubricated. And so it's actually a combination of these guys. These are the major two, that's why I've got them underlined. And these guys also will sometimes have problems. And this is technically the correct term for sty. A sty is actually a problem in the sweat glands. And these guys, these sties normally turn up. Can you see the location of this thing? They normally turn up right on the edge, and they look external. So, good illustrations for problems with people's eyes that you can see related to the glands and the muscles that are there. Droopy eyes, eyelid problems, and the real common things. Now, once we get inside the eye here, okay, you can see here in this picture, the inside part of the eyelids actually fold and become continuous with the outer surface of the eye. Now, this makes it appear, when you look at this picture, that that surface there will continue across the front of the eye. But that is not right. That is not true. This structure here, as you can see, is called the conjunctiva. You've heard the term conjunctivitis. It is actually a vascularized layer on the eye. Now, when I taught you this in the fall, I said the corneas do not have any vascularization. That's why they need the aqueous humor, and I'm going to show you that again here in just a moment. But as it turns out, you can look in somebody's eyes who have a problem in their eyes, and you can see these blood vessels. And maybe you thought to yourself, well, Warren told us that there are no blood vessels in there. And so let's be careful, right? The conjunctiva is vascularized. That's where the blood vessels are that you see when you think of the reddening of the eye there, that conjunctiva. And it ends at the cornea. The conjunctiva does not cross the cornea. And so the cornea becomes an area, a clear area. The conjunctiva is not clear and vascularized. The cornea is not vascularized. So I did not tell you anything wrong. I just didn't finish the story. Now this conjunctiva inside here serves as a pocket where the contact lenses are. People have asked me before, students have come up to me and said, what happens if I rub my contact lens? Can it go around inside my brain? And that's just because they haven't had enough anatomy to learn about the conjunctiva that attaches near the border of the cornea, crosses the white of the eye, the sclera, makes a fold and connects to the inner portion of the eyelid where they change names there. Bulbar versus conjunctival anatomy pieces there. All right, so I mentioned the conjunctiva, I mentioned the eyelids, and I think at this point we can make our way in. But before we do, let me remind you, the major watery secretion that is produced in the eyes by a gland called the lacrimal gland. The secretions of this lacrimal gland cross the surface of the eye here and are drained by small openings on the medial aspect of the eye called puncta, or singular punctum. They then drain on the medial aspect of the eye into what's called a lacrimal sac. And then that lacrimal sac then empties, as you remember from the fall, near the inferior meatus, near the border of the inferior nasal concha inside the nose. Now, if you're wondering, this portion of the eye here, not labeled, this is the caruncle here that we pointed out in anatomy, also has a dense concentration of sebaceous glands there. Now, you guys knew this because those sebaceous glands will oftentimes build up sebum that will dry out during the night, and you wake up and you have material in your eye. That's dried, oily sebum from this caruncle. It's also interesting to note there that the inside of this duct system here that drains into the nose obviously has a mucosa that's continuous with the inside lining of your nose, which means infectious material can actually make its way up this duct system. And oftentimes, when we have a nasal congestion, right, because knows where I'm going with this, oftentimes when you have a nasal congestion, something wrong with the lining of the mucosa, it will cause inflammation within these ducts, within these puncta system, and then what happens? Your eyes water. That's lacrimal gland secretions that are unable to make their way into the drainage system. We also know that when we are crying, and there is an overproduction of tears from the lacrimal gland, that the puncta cannot handle the volume, and the tears then spill out over the palpebrae onto our cheeks. It's interesting. It's an interesting philosophical question as to why human eyes make tears. Why would emotion cause an increase in the amount of tear production? Even the Merritt text, if I remember correctly, says something like, the reason for the production of tears is poorly understood. That normally means we have no idea. All right. Now, the muscles that move the eyes, before we go internally the eyes, remember there are 6 extrinsic eye muscles. The superior, middle, inferior, and lateral rectus muscles, and then the obliques. And remember your famous LR6SO43 and the motions of these eyes. I will ask you again to remember this. It's important. It will never go away. You're talking about what the eyes do. This is important information. The functions of these muscles that are attached to the eyes, moving them about. Interesting, I didn't talk more detail on this, but there's a lot more to be said about the planes that the eyes move in that you'll learn, I think, probably later in your life. I'm not going to take the time to do it here because it takes a while to describe it. But I wonder, did you wonder in the fall to yourself, okay, this moves the eye lateral, this moves it medial, this elevates it and this depresses it. Why do we need oblique muscles? Can I not elevate the eye without the oblique? I can. Why couldn't I just do a combination of rectus muscles with this function instead of having an oblique muscle? Can I not turn the eye lateral? Can I not depress the eye and turn it lateral? What's the point then? What is the point of having these? And it's really all about the angles in the eyes, because the way these muscles attach to that annular ring, remember that ring in the back of the eye? The way they attach to that ring is from lateral to medial, mostly. Which means the tendency for these rectus groups, the tendency when they contract is actually a medial turn in the eye. That is the tendency. So that is, in my opinion, that's the best. The reason I bring it up is to just try to help you remember. This is hard to remember. This lateral motion here is, you memorized it, but if I give you something to remember by, these muscles all tend to have a medial function because they all move medial from the annular ring. Now the lateral rectus is obviously not that way, but the other three at least have a tendency to want to move the eye medially. These oblique muscles are not just for elevation and depression. They give the eye a balance. So we use them whenever we move our eye at all. We move our eyes at all, we use them in order to help us make a more lateral impression, a more lateral movement for the eye. These are also in a different plane, I know, but I just wanted to try to give you something here. I can remember this always because inferior makes me think it would move inferior, but it's the opposite. And this one, superior, makes me think it was superior, but it's the opposite. So I don't forget that. This is the one that tends to forget here. It has a lateral motion. The oblique muscles tend to move the eye in a lateral fashion to avoid the problem with the attachments of the rectus moving it medially. LR6, SO4, 3, the innervations. All right, now, light, as it turns out, is an incredibly interesting phenomenon. Visible light sits in the electromagnetic spectrum between 400 and 700 nanometers. The Roy G. Biv that you guys learned in physics. But light is interesting not just because it's energy, and not just because it works like a wave, but it's also interesting because light also behaves as a particle. Yeah. And so the interesting things to be said about how light does what it does in our eyes just goes on and on and on. Now, so we can take that from either perspective. What I'm going to do with you guys is I'm going to use the wavelengths here to teach you about the cones. So the short and the medium and the long cones and the wavelengths that they mostly absorb in here. But I like to bring it up at the beginning here that light has multiple properties. It's not just energy. It also has a particle nature, which is a very difficult thing conceptually for me. Very difficult to work through. Okay, so just the 400 to 700, we're going to come back to that with the wavelengths and the cones. All right, but let's work our way through. For the light, talk about some nervous system stuff here. Light passes through the cornea. You're aware of this already. On its way to the retina. Now, as light passes through this cornea, it passes through liquid that's present here in a cavity called the anterior segment, or the anterior chamber of the anterior cavity. And then the light passes through an opening here called the pupil that is surrounded by a piece of smooth muscle called the iris. This iris exists in two distinctly different looking, arranged smooth muscle patterns. A constrictor around the opening and radial around the edges. So I want to show you that and talk about its innervation once again in some detail. And I also want to review with you very briefly the movement of this fluid and where it comes from. This fluid is called aqueous humor. It is produced in the middle layer of the eye. Remember, the ciliary body produces this fluid, releases it into the chamber called the posterior segment or posterior chamber of the anterior cavity. Remember, this back area here is the posterior cavity, and we call these chambers of cavities or segments. This chamber gets the fluid, it passes through the pupil, and then is drained by the scleral venous sinus at the edge of the corneal scleral junction, very near where the conjunctiva attaches. So I want to blow that up again and show it to you. First the muscles, and then we'll do the fluid. Blow each of those up. I just mentioned them, so let's talk about the details of both now. So this shows you what happens when light hits the eye, and the illustration here is intended to reteach you, again, the autonomic nervous system control of this. Notice that the pupil is surrounded by smooth muscle. There's a constrictor layer here, or a circular layer, and then there's a radial arranged layer on the outside of that. Notice that this shows you that information to the radial muscle comes from the superior cervical ganglion. Now the superior cervical ganglion is the top ganglion of the sympathetic chain. This is sympathetic information. What would be the neurotransmitter released by postganglionic sympathetic fibers on these radial smooth muscles? I keep telling you it's never going away, and eventually, maybe by April, you'll believe me. It's never going away. Never, never, never going away. It's going to come up and bring this stuff up every time we do a test. The radial smooth muscle here is innervated by the superior cervical ganglion. That is the top ganglion of the sympathetic chain, right? The top ganglion of the sympathetic chain. This one. You can see it innervating the iris here, this top chain. A chain of ganglia that lies along the spinal cord. Information coming from T1 to L2. The thoracolumbar division of the sympathetic nervous system. Information coming here. Postganglionic sympathetic fibers release a neurotransmitter called norepinephrine. Norepinephrine. And what is the receptor type? Adrenergic. Okay. So whenever these guys contract, whenever these guys get information from the sympathetic nervous system, they stimulate, cause excitatory postsynaptic potentials in the radial smooth muscle. The adrenergic binding does. What happens to the pupil size? Wait. It increases. That causes dilation. That's right. So the information, the postganglionic sympathetic information, causes dilation of the pupils because of this. So drugs that are used to dilate the pupils are adrenergic agonists. Right? Okay? Now, we also know that the inside layer of smooth muscle is innervated by postganglionic parasympathetic things. Which cranial nerves are used to deliver parasympathetic information? 3, 7, 9, and 10. Which of those cranial nerves goes to the eyes? 3. Right. So in the parasympathetic piece here, you can see shown up here, cranial nerve 3 and its innervation of the eyes. Specifically, what part of the eye here are we talking about? Well, we're talking about the constrictor layer. What's the receptor type? What is the neurotransmitter released by postganglionic parasympathetics? What is it? Acetylcholine. What's the receptor type? Muscarinic. Cholinergic. Okay, so that would cause constriction of the pupil. And this is once again the importance of the autonomic nervous system. This is regulating the amount of light. Now, can any of you guys remember the answer to the question, why is it that when you shine light into one eye, that both eyes constrict? Where does that happen? No, there is crossing of fibers of the optic chiasm. I'll put the picture in to go through with you later in the presentation. I was hoping that you would remember from the fall. What if I said pretectum or Westfall? Does that ring any bells? Okay, so we'll look at that again as we get to it. I'd like for you to be able to answer that question once again in the spring. And then I want you to see it for yourself in the lab next week. So one of the things we're going to do is have a few visual experiments to do in the laboratory. I'm also going to ask you, I'm going to talk you through the saccadic eye movements in this class before next Thursday. And then next Thursday, I'm going to ask you to hook your eyes up and watch what happens. Watch what your eyes do when you read. It's fascinating to see what your eyes do while you read. You can actually see word to word to word reflected in the saccadic motion of your eyes while you read on the computer screen. We're going to hook your eyes up. I'm going to give you a sentence to read, and then as you will read the sentence, and then you will look at the computer screen and you will be able to count the number of words on that line related to the motion of your eyes moving from word to word. It's fascinating stuff. Okay. Now, I also want to review with you the aqueous humor stuff. So, again, remember the ciliary body, this is the middle tunic of the eye, produces this aqueous humor into what's called the posterior chamber of the anterior cavity or segment. And then the fluid goes through the pupil into the anterior chamber and then is drained through the sclerovenous sinus. So this venous sinus is a drainage location for the aqueous humor produced. That is being produced all the time. What happens if the sclerovenous sinus does not drain aqueous humor properly? Pressure in the eye builds up. It's called intraocular pressure, or IOP, in the clinic. And what is the clinical condition? Glaucoma. Yeah, glaucoma. So everybody gets tested for the function of the sclerovenous sinus whenever you go and get your eyes examined. All right. Now, this is just another blow-up of that picture to illustrate the ciliary body and the difference in the muscle. This is intended to help me introduce as the next concept here is we're controlling the light entry into the eye. The next concept here is going to be how the lens actually modifies how light passes through the eye. So I want to talk about that with you in detail. So light has passed through the cornea, through the aqueous humor and the pupil, and you've been able to track the events that are occurring here. You can now think, I'm hoping, you can now think about your eyes opening and closing and the nerves that do that. The nerves and muscles that move your eyes, you can do that. You can talk about problems with the liquid on the surface of your eyes. And now you can talk about problems with the liquid inside your eyes. And now, as we're moving light through here, we want to talk about issues of controlling the focal point of light that's passing through the eye on the retina. And that is done by bending the light. So refraction is done mostly in the cornea. You have to bend the light as it passes through the cornea. You have to bend it so that it reaches a focal point on the retina. Otherwise, you don't see clearly. And one of the ways we focus ourselves, we focus the light on near and far vision, is by changing the shape of this structure that sits just behind the posterior chamber, just behind the pupil, a structure called the lens, which is supposed to be clear. It's not on any of the eyes that you guys do in the lab. It's not clear, but it's supposed to be clear. If it's not clear, that is also a clinical condition that we call cataracts. Yeah, some cataracts. So this lens needs to be clear, and it's very helpful if the lens material is pliable. As it turns out, as we get older, this structure loses some of its elasticity. And as it does, we lose some of the ability to focus for near and far vision, and we end up with bifocals because of that. And so the loss of elasticity of the lens also results in a clinical condition known as, I'll give you these terms again here momentarily. The loss of elasticity is known as presbyopia. Okay, so we'll do that one again here in just a second. All right, so the lens has to, okay, so what does it do? All right, so the lens is attached, as you saw in this picture. The lens is attached by way of, they're oftentimes referred to as zonular fibers or suspensory ligaments. You can see these guys are attached over here to the ciliary process. And notice that there is muscle here. Now, does that muscle right there in that ciliary process look like skeletal muscle to you or smooth muscle?
It's skeletal.
It does? What does it look? I mean, I'm not asking you to know what it is. I'm asking you what it looks like. It looks like skeletal muscle, yeah? And so we learned in anatomy the difference between single unit and multi-unit smooth muscle, right? Can you help me with that? Single unit and multi-unit smooth muscle? So the most common type is a single unit muscle, and they had varicosities over them, gap junctions between them, and they'd be like sheets. But these guys here, they actually operate more like skeletal muscles. You don't have the gap junctions. They operate as individual cells, and the varicosities go to individual cells. They're more controlled in the way you would think a skeletal muscle would be controlled, but they're not. They're smooth muscle. The ciliary muscle is smooth muscle, and it is controlled by cranial nerve 3. Right. So this nerve is also responsible for changing the shape of the lens. Cranial nerve 3 is. And so the lens changes shape like this. Whenever we want to see something that is distant, we relax the ciliary muscle, which causes the suspensory ligaments to become tight and the lens to become thin. Focusing for far vision allows for the thinning or flattening. It makes for the flattening. This is, in my mind, I always get this backwards because I think if I want to do something, if I want to look at something out there, I want to use it. But in this case, it's just the opposite. And you think, well, in this case, I'm letting the lens be what it is. And that's not true either. Because as these fibers relax, it actually pulls on the lens. The more natural, unchanged shape of the lens is when we look close up. It's interesting. So when we look far off, it's like you've got to pull that thing out flat in order to focus on things that are far off. And it's interesting because this is something also I'll ask you to do in the lab and read about in your lab manual. At 20 feet, this is where the switch is. So if you get closer than 20 feet, you're slightly contracted with the muscle. If you get further away than 20 feet, you're becoming relaxed enough to pull on the suspensory ligaments. But at 20 feet is where you're neither pushing nor pulling. And so that's why an eye exam is set up to use that distance in order to test the function of your eyes. So that you've sort of taken the lens out of the picture. You follow that? The lens is not really in the picture if you're neither stretching it nor if you're neither thinning it nor rounding it. All right. So memorize these: relaxed, taut, thin, far vision. Contracted relaxed ligaments thickened for close vision. Why are my eyes tired after I've been reading for hours?
Because they've been contracted.
Because the muscles have been contracting, right? The ciliary muscles have been contracting to do that. All right, now here's some clinical terms for you and answers to the questions what these are and how you fix them. Now the first one I mentioned already is not on this picture, is presbyopia, P-R-E-S-B-Y-O-P-I-A, is the phenomenon caused by the inelasticity of the lens. Now these are much more common, well, not more common, these are very common terminologies used to describe nearsighted, farsightedness. I expect you to know what they mean. All right, so first, an emmetropic eye, or emmetropia, is normal. A normal eye has the correct distance from the cornea to the retina, so that the focal point caused by the bending of the light, primarily by the cornea and the lens, will actually focus on the retinal surface. If this is true, you do not have a visual problem. If you are nearsighted, the problem is your eye is too long. You can see here in the picture, the eye is too long so that the bending or refraction of light in the cornea and the retina primarily causes a focal point to occur in front of the retina. Now, in order to do this, you have to cause a divergence. You have to diverge. You don't want to converge, you want to diverge the bending patterns, and we do that with concavity. So concave lenses are the way of diverging light, and this is what your eye doctor is prescribing for you with your negatives and your positives. I'm going to hand you an ophthalmoscope and let you see it for yourself. You can put the negatives and the positives on the ophthalmoscopes that I have and predict whether the person is near or farsighted, assuming your eyes are normal. I'm going to teach you how to do that in the lab. This is myopia or nearsightedness. Hyperopia is farsightedness, and you can see just the opposite. The eyes are too short, and so the focal point actually ends up behind the eyes. So in this case, you want to converge. And so the fix here is a convexity. And then finally, everyone has astigmatism. We normally use the term clinically, astigmatism. It actually means there's an irregular curvature. Astigmatism means the curve is not perfect, which means the bending happens. You can see here the faulty bending here of the light so that you do not get a focal point here caused by these. The truth is, no one has a perfect eye. So normally when we say in clinical settings today you have astigmatism, well, what we mean is you have a curve that is sufficiently large enough to create a visual problem. And so these guys can be corrected in a number of different ways. Glasses can certainly do it, but sometimes lenses that have no convexity or concavity can be placed on the surface of the eye and fix this. So just to press, some people wear lenses at nighttime with a thickening on one side of the lens or the other. Gravity helps to put that lens where you just put it in your eye, and it orients where it's supposed to be because of the bending, because of the curve of the eye, and then it's designed to sort of press on the stuff. You wake up the next morning, take the lens out, you're fine for the day. Put it in at night, do it again. Some people have lenses that they wear permanently to fix astigmatisms like that, with just a thickened portion on one side or the other. So, or glasses, like I said, a number of different ways to fix the problem with the bending caused by the irregular. What is an astigmatism? An irregular curve in the cornea causing the problem of bending. Presbyopia, emmetropia, myopia, hyperopia, and astigmatism. These are very common terms. Every It's all right. Every physiology kid needs to be familiar with it. Let's see the screen.
I dropped the order.
You have?
Yeah.
I want to see.
Oh yeah. That hurts my heart.
It hurts my heart. You taking your foot and dragging it.
How is it so long?
Oh, because
She doesn't have
Macintosh takes a licking and keeps on digging.
None of the parts
Must be
move,
nice.
so they don't break it.
Okay. All right. So once light gets to the retina, so we've moved light through and we talked about the structures light's going through. Once light gets to the retina, if we're going to focus on the light in the retina, we normally do so for visual acuity purposes on a structure called the fovea centralis. Now, you can use the ophthalmoscopes that I'm going to give you to see if you can find this. I am no good at this. I've had eye doctors here. Dr. Robinson used to come every year. I've taken kids to the clinic, to Dr. Redmond's clinic for years. I've had him do it. I have ophthalmoscopes, and so I'm just no good at finding the structure. On pictures like this, you think, well, it looks so obvious. I think I did. So you look through here, this is, let me turn the lights off. Can you get the lights there?
Switch over there.
Oh, great. So this is actually a picture of a diabetic eye here, and you can see capillaries that have major blood vessel problems here. But can you see the little darkened area here, the macula? This is temporal and nasal. So which eye? So this is a right eye, but you would know that if you look into an eye, you can tell immediately which one is right, which one is left, because this structure you can see really easy. Even if your eyes are not dilated, you guys are looking at each other's eyes in the lab. When you shine the light in there, guess what's going to happen? It's going to constrict, make it very difficult to see. And so I always laugh when I'm at the eye doctor. I go to this place over here once every 5 years faithfully by the, what's it called? It's the place where they get the really cheap glasses. Not so cheap.
Walmart.
Yeah, it's over there by the AT&T store.
Oh, Vision for Life.
That's the
That
one.
place.
Yeah, so I go in there and you use that place too. And the guy's so nice. The reason I go in there is because it takes him like 3 minutes and we're out of there. I don't want to mess with it. And so he comes and he puts that light in front of your eye. And then he says, keep your eye open there. And then he goes, moves it side to side. I'm going, you're not seeing anything. I'm not stupid. When you shine the light in there, it's constricted. So when you move that thing this way, you're not seeing anything. You're not tricking me. You're acting like you're doing a full eye exam, but you're not. The only way, I don't say that. I don't say that to you. I just want him to put the things in front of me and go, this one or this one, this one or this one. Okay, give me some glasses and I'll go away. But it's very difficult. The point there is to say it's very difficult to see this macula, which is where visual acuity happens. Now, is this a left or a right eye? That's a left, yeah. So this place here is called the optic disc, and this is where all the optic nerves lead. It's also where the blood supply comes in. Most of these blood vessels are actually in the choroid coat, the middle layer, the vascular layer, and you're actually looking through the retina here at the blood vessels within the choroid coat. And you can see what diabetes does to the ends of these capillaries within the diabetic eye. It makes a real mess of things. This person is having all kinds of visual problems. I wouldn't be surprised if there was partial blindness here. But the reason for the picture here really is to illustrate the importance of this area, the macula lutea, which is supposedly a yellowed area of the retina. And in the center of this macula is a little indention. It looks like somebody touched it with a pencil. There's a little indention there called the fovea centralis. And I was hoping from anatomy you remember this part about the fovea centralis. I told you guys that there are about 120 million rods in the retina and about 6 million cones. So we are rod photoreceptor-oriented creatures. And rods are mostly for gray scale determination, edges of the night and day, which is interesting. Cones are really for color vision, and we're going to talk about the medium. We're going to talk about the short, medium, and long cones, which I did not do in the fall. Remember that there are over 1 million axons. So in the retina, let's review this just very briefly. In the retina, remember, As light passes through, it passes through a ganglion cell layer, then a bipolar cell layer that has amacrine cells between them, which we're not going to talk about any further. That is interesting. You see how they're connected to the ganglion cells? And this bipolar cell layer is connected to what we call a photoreceptor cell layer. And you can see those horizontal cells that are connecting those. And so you can see this picture illustrates a lot more rods than cones. And so you can also see that the rod cells here in this picture tend to be connected, many rods connected to a single bipolar cell, and many bipolar cells connected to a single ganglion cell. And then the ganglion cell axons are actually the optic nerve. This is where the information is. So the light comes through, it has to hit the photoreceptor, it does something. That's physiology. We're going to do that, right? When the light hits the photoreceptor, that's when vision happens. All this other business is mechanical compared to the phenomenon that occurs now when this happens. When the light hits the photoreceptor, now vision is underway. And then the visual signal comes this way, which is backwards, according to everything you've learned so far about neurophysiology. What we're going to see is that in the dark, these guys are stimulating these guys, which are then in turn inhibiting these. You see how that's backwards? You think, well, my eyes are open. That would cause stimulus. But instead, when your eyes open, it shuts these down. So for some reason, the bipolar cells want to stimulate. In fact, if you measure the action potentials in the optic nerve, there's always a low level of action potential coming down the optic nerve, even when your eyes are closed. So that's a bizarre phenomenon that occurs here. Okay, but what I wanted to do here first is talk about the numbers. The numbers, this is a fascinating phenomenon. The numbers are about, in terms of the number of photoreceptors and the number of axons. So these are ganglion cell axons, about 100 to 1, which means that each ganglion cell, on average, is an accumulation of information from over 100 cells, 100 photoreceptors. Massive amounts of convergence. I use that to really drive home the point that this area that I've been talking about, the fovea centralis, is very unique. There are about 4,000 photoreceptors there, and there are about 4,000 ganglion cells. That explains the phenomenon. Why do we move our eyes in such a way to focus on this spot? We put the focal point here. When somebody has macular degeneration, they have a visual acuity problem because that's where visual acuity happens, in the place where the ratio of photoreceptors to ganglion cells is 1 to 1. 4,000 to 4,000. Now I'm going to talk some more about this phenomenon in the saccadic eye motion thing here with the fovea in just a moment. Probably not today.
So, like my color blindness, is that, a problem with cones? I'm not, cones.
It is. So you give me just a second. I'm going to do that with you. You're right on target here, right? Okay, so we know that there's a couple of different kinds of photoreceptors. We've seen that before. We named them really for the shapes of their outer segments. You can see these are rod shaped and these are cone shaped. Now, the rod cells, yes, for low light vision. Cone cells we think of as being for two things. We think of them as being for what we just said, for visual acuity. Now, can you explain to someone why we say cones are for visual acuity?
Because there's more cones in the fovea.
Right, because the density of cones is in one spot in particular. There are cones in other places, but the density is in the fovea. And why is it that the fovea now tells me that the cones are for acuity?
Because that's where your eyes focus on.
Yes, because of the ratio of cell to cell, right. So there's no convergence of information here. The light information that occurs in this cell is the light information the ganglion cell sees. That is the answer to the question for acuity. Okay, the second question then is color. And we say cones are for color vision. Okay, and the primary reason for that, oh yeah, I put that in. The primary reason for that is because when you're measuring the wavelength absorption for these cone cells, you find that that there are 3 general varieties of cones. Okay, so how does a physiologist, how does a neuro person, how does a visual specialist researcher come to know that there are 3 different kinds of cones? Can you look at them? Can you look at these guys? Is this an S cone, an M cone, or an L cone? How would you know?
If you show like one color and it start to like use that, you see what's used more, and then
If it, I guess if it goes to the information goes to the ganglion cell, you'll describe it.
Okay, so some kind of sophisticated experiment to isolate the ganglion cell. No way to do that. That would be a good way to do it, but how would you experimentally isolate one of these ganglion cells? I mean, that would be, there's got to be another way that we would know. The molecular biologists are going to have to give us some help here.
well, when I look at like screen, like if I look at one color for a while, it kind of bleaches that color.
Okay, good. Yeah, you're all getting around it, but the point is this. It's the type of proteins that are in these cells. And now the analysis can be done really easily, just using a Western blot, because the proteins have different sizes. So we know from general experiments like this with different colors that you have these 3 abilities. But you can actually isolate individual cone cells now and tell which ones they are. In fact, we now know, as this picture indicates to you here, I think it does. Yeah, we now know the chromosomes where each of these proteins, we call them photopsins, that are produced for the cone cells are actually made. And so, show the picture and then go back to it. So, we know that there is one population of cones that have proteins in them that absorb best in the blue. We know that there's another that absorbs best in the green, and we know there's another that absorbs best in the red. So, we say that cones exist in the retina in these 3 varieties: blues and greens and reds. So the numbers are 420 for the blues. That's the best. That's the top of the spectrum. Does it not absorb anywhere else? It does. Sure. It has a width of spectrum. This is the peak of the spectrum. This is the short cones. 530 for the greens. These are called the M cones. And 562 for the reds, are the L cones. Now, notice I've got here the M's and the L's are along of the X chromosome. If I told you that 8% of the male population has either red or green color blindness, protan or deutan color blindness. In fact, the most common color blindness is green, a problem with the medium cones. That's the most common in the population. If I told you that 8% of males and less than 0.5% of the female population has this problem, does it help you to know that the M's and the L's are on the long arm of the X chromosome? Why? Why do guys have color blindness more than girls? We don't have one X. We don't have one X. So the girls have a much higher. So we can't. It's more difficult for males to carry the recessive condition because of that. So 530 on the greens, 562 on the L's, the M's and L's on the long arm. Now, at this point, I'm going to begin to talk about some details in visual fields and the way vision actually occurs. It's going to get a little sticky. Okay, we're going to do some molecular biology, but as it turns out, this is the kind of information that you have to know now to be able to describe vision. Vision, from what I've told you so far, we've really not done vision yet. Because vision, as you guys know already, vision is a phenomenon that occurs in the occipital cortex, right? A phenomenon that occurs in the superior colliculus. If you have damage to your brain, your eyes can work just fine. You're not going to see. So what we're going to do on Wednesday, what we're going to do on Friday is I'm going to try to answer the question for you. What makes the action potentials in the ganglion cell axons? Now, for your interest and for your enjoyment, we're going to walk through this detail, all right? For your interest and enjoyment, this gentleman, I think probably most of you are familiar with. If you've had Criswell or Shank, they introduced Behe and his famous landmark publication, Darwin's Black Box. So he uses vision to talk about stuff there. This guy is very dear to me. He is currently the head of the biology department at the University of Kentucky. He was my graduate advisor, Vincent Casson. He was also the head of the Department of Biology at Texas A&M for several years. And while he was there at Texas A&M, Michael Behe came to campus, and they had a forum to talk about Behe's Black Box in Rudder Auditorium. There were thousands of people there. And I was not in attendance. I'd already graduated and gone on, but of course I knew this event was happening. I don't know this guy, but I've read his work. I know this guy, and I know everything that he thinks. And the conversation between these two about irreducible complexity is one of the most thorough that exists online. It's a long one, and I'm not asking you to watch the whole thing. It's a lot here, but I welcome. I put the link up. I checked it from last year. I checked it to make sure it's still there. And so if you're interested, this is for your enjoyment. One day when you have a lot of time, you're just sitting around and you want to listen to these guys talk about irreducible complexity. Vision comes up as part of the discussion. I recommend, and you can think about me while you're listening to it. Yeah? Because I don't know him, but I agree with him. I know him and love him, but I disagree with him. So these two guys talking is, I'm fascinated by it because I'm so, closely associated with these people. And I like to make students aware of it. I'm not going to make you watch it, but at least you're aware.
So Casson is an evolutionist?
He is indeed. All right, so visual physiology on Friday.